Good morning. Today we are discussing about this enzyme specificity, how enzymes are specified. Enzymes are highly specific in their action when compared with the chemical catalysis. The occurrence of the thousands of enzymes in the biological system might be due to the specific nature of enzymes. There are three types of enzyme specificity are well recognized by the scientific community. One, stereospecificity. Second, reaction specificity. And third one is the substrate specificity. The specificity is a characteristic property of the active site of the enzyme. First, we have to be discussing about the stereospecificity, which is otherwise called as optical specificity. Stereoisomers are the compounds which have the same molecular formula but differ in their structural configuration. The enzyme acts only on the one isomer and therefore which exhibit stereospecificity. For example, L-amino acid oxidase and D-amino acid oxidase act on L and D-amino acid respectively. Consider L-amino acid is acted by L-amino acid oxidase, which is deaminated to corresponding keto acid, and ammonia is released out or excreted out. The hexokinase act on D-hexose. Glucokinase act on glucose. Hexokinase, which is considered to be a common enzyme which act on all hexoses except galactose, whereas glucokinase is a specific enzyme which acts only on glucose. Amylase act on alpha glycosidic linkage. Cellulase act on beta glycosidic bond. The second one is the reaction specificity. The same substrate can undergo different types of reactions, each catalyzed by the separate enzyme, and this is referred as the reaction specificity. An amino acid can undergo transamination reaction, oxidative deamination, decarboxylation, racemization, etc. The enzyme, however, are different for each of these reactions. Substrate specificity means the substrate specificity varies from enzyme to enzyme. It may be either absolute, relative, or broad specificity. Absolute specificity means certain enzyme act on only one substrate, that is glucokinase act on glucose which gives glucose 6-phosphate. Urease cleave urea to ammonia and carbon dioxide. The second is relative specificity. Some of the enzymes act on structurally related substances. This in turn depends on the specific group or bond present. The action of trypsin is a good example for group specificity. Trypsin hydrolyzes peptides or peptide linkages involving the basic amino acid arginine or lysine. Chymotrypsin, which is the one among the proteolytic enzyme, which cleaves peptide bond attached to the aromatic amino acid phenylalanine, tyrosine, and tryptophan. Example for bond specificity: glycoxylases acting on glycosidic bond of carbohydrates, lipases cleave ester bond of lipids, etc. Broad specificity, or what can call as the bond specificity, some enzymes act on closely related substrates, which is commonly known as broad substrate specificity. For example, hexokinase act on glucose, fructose, mannose, and galactosamine, not on galactose. It is possible that some structurally similarity among the first 4 compounds makes them a common substrate for the enzyme hexokinase. And we have to discuss more about the coenzymes. Coenzymes are non-protein organic low molecular weight and dialysable substances associated with the enzyme function, which is called as coenzyme. The protein part of the enzyme on its own is not always adequate to bring about the catalytic activity. Many enzymes require certain non-protein small additional factors, which are collectively referred as the cofactors or for catalysis. The cofactors may be organic or inorganic in nature. The functional enzyme, which is referred as the holoenzyme, which is made up of protein part, that is otherwise called as apoenzyme, and a non-protein part, which is called as a coenzyme. The term prosthetic group is used when a non-protein moiety is tightly bound with the enzyme, which is not easily separatable by dialysis. The term activator is referred to inorganic cofactors like calcium, that is divalent calcium, magnesium, and manganese, etc., which necessary to enhance the enzyme activity. Coenzymes are otherwise what we call as the second substrate. Coenzymes are often regarded as the second substrate or co-substrate, since they have affinity with the enzyme comparable with that of substrates. Coenzymes undergo alterations during the enzymatic reactions, which are later regenerated. Most of the B-complex vitamins are considered to be coenzymes. For example, consider vitamin thiamine is a coenzyme of considered to be thiamine pyrophosphate, that is TPP, acting on trans-ketolase enzyme. Vitamin riboflavin is a coenzyme that is for FMN, that is FMN monoflavin mononucleotide, acting on the riboflavin enzyme. Hydrogen and electron L-amino acid oxidase. Vitamin riboflavin coenzyme, that is FAD, act on riboflavin, and D-amino acid act on vitamin niacin is considered to be coenzyme for nicotinamide adenine dinucleotide, which is acting on lactate dehydrogenase. Vitamin biotin is a coenzyme for biocytin acting on the enzyme pyruvate dehydrogenase. And the coenzyme for B-complex vitamins are, most of the coenzymes are derivatives of water-soluble B-complex vitamin. In fact, the biochemical functions of B-complex vitamins are exerted through the respective coenzymes. Non-vitamin coenzymes also there, which are not at all coenzymes of vitamin derivatives. There are some other organic substances also, which have no relation with the vitamin, but function as coenzymes. They may be considered as the non-vitamin coenzymes. For example, consider ATP, CDP, UDP, etc. The thermodynamic factors of enzymatic reactions, which include isothermic, exothermic, and endothermic. There are three types also there. That is, one is isothermic, second is exothermic, and third is endothermic. The regulation of enzyme activities, which includes the biological system, the regulation of enzyme activities occurs at the different stages in the one or more of the following ways to activate or achieve the enzyme cellular economy. One, allosteric regulation. Second is activation of latent enzymes. Third is the compartmentalization of metabolic pathways. Fourth is control of enzyme synthesis. Fifth is enzyme degradation. And sixth is isoenzymes. Allosteric regulation and allosteric inhibition, which is clearly illustrated in the figure. Some of the enzymes possess additional site known as allosteric site besides the active sites. Such enzymes are known as allosteric enzymes. The allosteric sites are unique places of the enzyme molecule. Allosteric effect is also there. Certain substances referred to as allosteric modulators are called as modifiers, binds at the allosteric site and regulate the enzyme activity. The enzyme activity is increased when a positive allosteric effector binds at the allosteric site known as the activator site. On the other hand, the negative allosteric effector binds at the allosteric site called as the inhibitor site and which inhibit the enzyme activity. Another one is called as feedback regulation. That is the process of inhibiting the first step by the final end product is the series in the series of enzyme catalyzed reactions of the metabolic pathway, which is referred as the feedback regulation. Look at the series of reactions given below. A is converted to B by enzyme E1, B is converted to C by enzyme E2, C is converted to D by enzyme E3, D is converted to E by enzyme E4. So final product is E, which is fed back and which is inactivating or inhibiting the activity of the first enzyme E1, which is called as the feedback inhibition, or it may be considered to be feedback regulation. Feedback inhibition or the end product inhibition is a specialized type of allosteric inhibition which is necessary to control the metabolic pathways for efficient cellular function. So here we are shown a pathway which is regulating the urea cycle, that is carbamyl phosphate plus aspartate by the enzyme aspartate transaminase. That is fed back to cytidine triphosphate and which is inhibiting the effect or regulating the effect of aspartate transcarbamylase. Thank you.